Hello, my name is Brooke Vickers, and during the spring 2023 semester, I was in Dr. Smith's lab, and his lab mainly focuses on insect biodiversity and taxonomy. So I would say this is a research lab, just because they are ever-changing, and they are constantly doing more research and identifying species of the darkling beetle. Their biggest aspect is focusing on terrestrial parasites and understanding the harmful effects they can have on human health. Dr. Smith believes by collecting and identifying all these species types, all the specific types of species in a location, this can help understand the ecological services those insects provide and the role they play in an ecosystem. This was a list of people who I interviewed and also spent time with. Dr. Smith, Chris, Olivia, Amelia, Charla, Adam, and Martha. So Martha is actually picking up research from a 1960s professor, Kirby Brown. She's doing taxonomic revision on the darkling beetle genus Bilotheus. Not sure if I said that right. She does DNA work, which involves comparing current observations with earlier research on the species. And her end goal is producing a set chart for identifying the species. So basically a brand new dichotomous key that will help identify the species. And then Charla is doing revisions on the Bilothvagani genus group. These are fungal eaters. They feast on polyporels, which are a certain type of fungus. And then they move on to, they make little tunnels in the fungus and then move on to the next one when they're finished. Her research is the basic backbone to most entomology practices. Also, some things I learned from her is that researchers worldwide benefit from the simplicity and rapidness of barcodes and QR codes, which make identifying more accessible. So next, I interviewed Olivia and Chris. So Olivia is working on this tribe of beetles, also darkling beetles, and it is known as the talkies because they do this tapping noise with their rump, whatever you want to call it, and there's 4 distinct patterns. She plans on doing more research in her 3rd chapter. Her work includes DNA extractions, PCR, and Sanger, and then next generation sequencing. And then they are synomorphic, which means they are evolved from a common ancestor. And then Chris is the collections manager, so his work serves as a repository for scientific data and specimens. Some things I learned from him is there are about 4 different labels with that certain specimen, and those labels include the date they were collected, location, and collection, and then also little codes like a QR code that leads to further information about that species. Also, this is important for the reproducibility of certain experiments, which relies heavily on taxonomy. So next, I worked with Amelia, who is studying this subtribe of darkling beetles. This is a picture I just drew from the internet. It's not what she's actually working on or took, but it's a certain species of this tribe, and I thought this is a great picture. So she is researching the links between basically the gut and digestive system with different types of fungi. And her goal is to know whether that tribe may be considered monophyletic, so basically derived from one common ancestor. Okay, and this is basically different specimens that I got to take from a bigger collection and just kind of sort and organize, and I had the great opportunity of actually adding them into the collection in the park, which there's about over 1.3 million different types of species down there, so that was really great and cool to get to work down there for a little bit. And yeah, these were different species I got to identify and then organize. So, like I said, the overall grand challenge is understand that insects are the base of every food pyramid, and that helps emphasize the importance of being able to identify species and the roles they play in every ecosystem, along with understanding the ecological services they provide. And this is just some terminology and also different things I got to learn. So a holotype is the main specimen used for identification. That was a word that was thrown around a lot when I was conducting these interviews and also working down the perk. A lot of careers in taxonomy are starting to decline, unfortunately. So it's really important to expand your knowledge and maybe minor in different fields of study. So Charla, for instance, is studies phylogenetics and evolutionary biology. And also another person, Adam Rourke, who's also part of their lab, he works with microbiology and is really good at dissecting different insects with very pristine tools. And also a dissertation, which I both know is being thrown around a lot, is basically consists of 3 chapters, and that together form a story. So overall, I loved working in this lab. It was really interesting, and I can't wait to see what more I learn.